map of a bustling city, right? But you're stuck in this zoomed out satellite view. You can see the gray blocks of buildings, the general layout of the streets, maybe the broad boundaries of the neighborhoods, but that's literally it. You have absolutely no idea what's actually happening inside those buildings. Are they hospitals, coffee shops, crowded apartments? Well, that is exactly where traditional tissue imaging in biology has been stuck for decades. We desperately need a way to zoom in, to get down to that molecular street level, so we can see precisely which cells are interacting, what proteins they're producing, and exactly how they function in their specific spatial location.
Welcome to this explainer. Okay, let's dive into this. Today, we are unpacking the completely incredible field of spatial multimodal omics. We're going to explore how scientists are taking cutting-edge data capture and combining it with advanced artificial intelligence to build that highly detailed molecular Google Maps of human tissue. So, here is our roadmap for today. First, mapping the microscopic world. Then, strategies for data capture, followed by the 5 core computational tasks. After that, we'll look at fusing multimodal spatial data, then AI models decoding biology, and finally, we'll wrap up with the future of tissue mapping.
All right, let's kick things off with Section 1, Mapping the Microscopic World. Now, what we're looking at here is an H&E stained tissue sample. This is the absolute cornerstone of standard histology imaging. It's routinely performed in clinical practices all over the world, and it really serves as the foundation of medical diagnosis. I mean, if a pathologist is looking for cancer, this is very often what they're looking at. But returning to our metaphor for a second, this is our satellite map. It shows us the morphology, the shape and structure of the tissue, but it completely lacks molecular depth. It just can't tell us the specific gene expressions or the protein abundances that are actually driving a disease at that specific spot.
Let's move to Section 2 and see how this builds. Strategies for data capture.
To actually gather these different layers of data, researchers rely on a huge array of techniques. What's really interesting about the breakdown here is how it divides these methods. You basically have image-based methods, which use high-resolution microscopes to detect targeted RNA or proteins right in the tissue. And then you have NGS-based methods, that's next-generation sequencing, which use spatial barcodes to capture a much wider, often whole transcriptome view. The ultimate goal? To stack these different modalities, like RNA, proteins, and that H&E morphology, right on top of each other.
But wait, how do you actually capture multiple layers from the exact same physical piece of tissue? Well, researchers generally use 2 main strategies. The first is adjacent slices. Imagine slicing a loaf of bread, right? But very, very thinly. You put consecutive slices on different microscope slides, and maybe you test one for RNA and the next one for proteins. The problem with this is that you have to use really complex computer algorithms to perfectly align or co-register these separate slices back together. The alternative, though, is co-profiling. This is where you capture multiple molecular layers simultaneously on a single tissue section. Co-profiling is incredibly elegant because it inherently aligns the morphological and molecular data from the get-go, completely avoiding those tricky computational alignment issues.
Moving right along to Section 3, 5 core computational tasks. So to actually build this map, the source literature outlines 5 specific, core computational tasks that researchers have to tackle. They need to co-register the modalities, deconvolve the cell types, identify spatial domains, predict omics from standard images, and impute missing genes. Let's actually look a bit closer at 2 of the most critical ones here.
First up is deconvolution. Here is the big problem. Most sequencing-based spatial tech doesn't capture data from just one single cell at a time. The resolution is wider, meaning each data spot actually contains a mixture of several cells. Deconvolution is kind of like trying to figure out exactly who lives in a specific apartment building when you only have the census data for the entire city block. It's tough. Algorithms have to mathematically untangle this mixture to infer the exact cell type composition at each tiny spatial location, and they often use single-cell RNA sequencing data as a reference point to do it.
Then we have spatial domain identification. Once we know who lives where, we literally have to draw the neighborhood boundaries on our map. Spatial domains are contiguous regions in the tissue where cells share similar molecular patterns and jobs. By identifying these, we can map out critical biological microenvironments. Think of it like spotting a fibrotic niche in an organ, or identifying a hypoxic, oxygen-starved neighborhood hiding inside a tumor. Which brings us to Section 4, Fusing Multimodal Spatial Data. Once we have these distinct layers of tissue data, how do we actually merge them without creating just a blurry mess? And this brilliantly illustrates exactly how we answer that question. It shows the overarching architecture of how raw spatial modalities, the RNA, the proteins, the imaging, are actually fed into machine learning models to accomplish those downstream tasks, flowing through very specific fusion paths. Basically, researchers have three chronological choices for when to blend all this data together. Early fusion is exactly what it sounds like. You directly combine the raw input matrices from the different modalities before doing anything else. Intermediate fusion is a bit more nuanced. The model extracts key features from each separate modality first, and then concatenates those refined features in a shared latent space, which is essentially a shared mathematical language for the AI. Finally, late fusion lets independent machine learning models process each modality entirely on their own, and only at the very end are their individual predictions combined for a final output. And honestly, the right choice here depends entirely on the specific biological question you're trying to answer.
All right, Section 5, AI Models Decoding Biology.
When we really look under the hood of these algorithms, we see different architectures that are perfectly suited to the biology itself. On one side, we have graph neural networks, or GNNs. GNNs are amazing because they essentially treat the tissue like a social network. They aggregate features from a cell's direct neighbors, preserving the local spatial geometry. On the other side, we have vision transformers and CNNs. Transformers are wild because they don't just look at immediate neighbors. They use mechanisms called self-attention and positional encoding to look at the big picture. They find long-range dependencies across the entire tissue slice, identifying coordinated biological programs that span really wide areas.
So the crucial point here is that this absolutely isn't a one-size-fits-all situation. Researchers have built specific, recommended algorithmic families tailored for each unique cartography task. For instance, if you're doing cell-type deconvolution, you're going to want probabilistic modeling tools like stereoscope or Cell2location. But if you want to identify those spatial domains we talked about earlier, you switch gears to graph neural networks like SpatialGlue. Aligning messy adjacent slices, you're going to use optimal transport models. It is a highly specialized toolkit.
And finally, Section 6, the future of tissue mapping.
So why does all this matter? Well, the ultimate holy grail goal of this entire field is democratization. Right now, capturing spatial omics data is incredibly expensive and it's super rare in clinical settings. But standard H&E imaging, that's cheap and it's literally everywhere. By training these deep learning models on data sets where we have both the cheap image and the expensive molecular data, the AI actually learns the hidden connections between them. The goal is to eventually take a simple, low-cost histology slide, feed it into the AI, and have the model accurately predict the rich, underlying spatial transcriptomic and proteomic profiles. We're literally using AI to turn the satellite view into a street view entirely computationally. Which leaves us with a pretty thrilling question to ponder. As these multimodal algorithms become more sophisticated, uncovering the exact molecular neighborhoods of cancer, Alzheimer's, and autoimmune diseases, we are no longer just observing tissue, we are truly reading it. So, how long will it be until this Google Maps of biology isn't just a cool research tool, but the actual standard guide for everyday clinical diagnoses, unlocking a whole new era of highly precise, personalized medicine?